Hey guys, so I'm going to spend a couple minutes here walking through the microscope lab with you guys and showing you or reminding you, maybe if you've used microscopes before, a little bit about how the microscope works and the way that you're going to want to approach this lab. We are going to be looking at a few different things under the microscope together and individually next class. So I want you to be very familiar with the parts of the microscope and how they work and how to actually use one once you approach one in class. So the first thing you're going to do, you're going to turn the microscope on. It's going to make the light shine. This is the stage, right? So this is where the microscope slide goes. We have a bunch of slides, some that you're going to make and some that we've already prepared or the companies have already prepared for you. One thing I want to make known, a couple parts that you need to be aware of. Underneath this is the diaphragm. So if you look underneath the microscope, there is this circle dial, and it has a bunch of different apertures, which means it has a bunch of different openings that you can turn over here. See where my hand is over here? You can turn, and do you see how that rotates through and how the opening underneath increases in size, and then it goes back all the way down to the smallest. That one I know is pretty hard to see. So that would be like a setting of 1, and then 2, and then 3, and then 4, and see how they get bigger and bigger and bigger. The point of that, the diaphragm under here, is to control how much light is shining through the slide so that it controls how much light you have when you're looking through the eyepiece and looking at your specimen. Sometimes when you're looking at specimen, you need a lot of light, and honestly, sometimes you need very little light. It depends on what you're looking at and what you're trying to see in the detail. Typically, I would always start around a 3. There is a number on the side over here, and it's going to be hard to see on the video, but when you are looking at the microscope yourself, you will be able to see whether you're on setting 1, 2, 3, 4, or 5. So again, 5 is the setting that's going to give you the most light, and 1 is the setting that's going to give you the least light. So you're always going to want to play around with that a little bit. You have your revolving nose piece here. So remember, this is what's going to turn your objective lenses through. This is a compound light microscope. So there's the light. That's obvious. Compound meaning there's a lens in here. So there's magnification power in the eyepiece. It's always a magnification of 10. And then you have another light lens here. So that's the compound, 2 lenses. You have a scanning or a low power, and then you have a medium, and then you have a high. Some microscopes have 4 objective lenses. Ours are all the same. They always have 3. So if you always want to start on the lowest magnification. So this is a magnification of 4, and this is a magnification of 10. So total magnification would be 10 times 4 is 40. You always start here, okay? Every single time. So always make sure that your revolving nose piece has been adjusted so that you're back at scanning. You take your slide. You're going to place it on the stage. These are the stage clips here, right? So I'm going to hold this up for you so you can see what I'm doing. So these are the stage clips. So you're going to place your slide so that the specimen is over the hole in the stage so that the light can actually get through, and you're going to move your stage clips over to hold the slide in place. Okay, so you're going to do both sides. Okay, these microscopes were just serviced, every single one of them. So the stage clips should be nice and tight. Everything should be really clean and nice for you guys to use. So once you have your slide in place, you're going to use one eye, and you're going to look through the eyepiece, and you're just going to try to find your specimen. You're going to move. Notice how I'm moving the slide around. Sometimes your magnification power or what you can see is really blurry. That's when you have to focus the images. This is called the coarse adjustment. This is called the fine adjustment. You always start here when you're on scanning power. You have to be really, really careful with what you're doing. If you're ever on medium or high, you cannot use this. This, watch what happens. Look at how much the stage moves when I turn this dial. So that's all the way down, that's all the way up. If you had a higher power objective lens down here, when you move the stage up, you're either going to break the lens, which is really, really expensive, or you're going to break the slide. Not as expensive, but still a problem, okay? Sorry, there's your bell. So please make sure you're only using this coarse adjustment knob when you're on the scanning objective. So when you're looking through, you're really just going to make sure visibly on the stage that the slide is centered over the opening, and then you're going to look through and you're going to find a specimen. It might be really blurry, so then you use this to adjust, and you just go really, really small, okay, until you find it nice and clear for you. Everyone has different eyesight, so that's going to be different for everybody. If you're wearing glasses or contacts, don't take them off. You wouldn't take your contacts out, but don't take your glasses off. You need them. But as other people are coming to the same microscope, you may have to make some adjustments, okay? So be aware of that. Also, really focus on having the specimen that you're looking at in the center of your field of view. The field of view is the circle that you see when you look through the microscope. So you want it right in the center, because as you increase your magnification with your lenses, all that's doing is narrowing your field of view. So if your field of view is here and your specimen's all the way out on the edge, when you're at this level, when you zoom in, you're going to lose it. And then you're going to freak out and you're going to ask me to help you. You don't need me to help you, okay? Get the specimen in the center of your field of view when you're here, so that when you increase your magnification, you're not going to have a hard time finding it again, okay? So once you're zoomed in and close, now you're going to increase your magnification. So you're going to turn the revolving nose piece to get to the medium or the yellow objective. This has a magnification of 10. This has a magnification of, remember, 10. So 10 times 10 is 100. So now you have 100 times the magnification of what you're looking at. So you look through again. I can already tell you that it's a little bit blurry. I'm not touching this, okay? Because this is going to make it even more blurry, because I don't have the fine motor skills to make the small adjustment I need here. So now I go to this guy. This is the fine adjustment knob. So I just turn him a little bit. Oh, that's the wrong way. So I turn him the other way, and I get focused, okay? In this case, I am gonna wanna move my slide a little bit. I know you don't see what I'm looking at, but basically I wanna make sure that, again, my specimen's in the center of my field of view, because he was just a little bit off to the side. And the other thing to think about when you get to this level, remember, we're looking at cells, we're looking at organisms in some cases, and these are three-dimensional objects, right? They're three-dimensional organisms, but when we make a slide out of them, we are pressing them down. So all of that volume and all of that filling of those cells and organisms is getting smushed, which means there are layers upon layers upon layers that are smushed underneath that slide and that cover slip. So when you're looking at it in the microscope, the problem is that some parts of the cell sometimes seem blurry than other parts. And so a lot of times as you're drawing what you're seeing, you have to adjust a little bit to see different parts of the cell so that they're in focus for you when you're drawing. You really have to take your time when you're drawing. A lot of my students sometimes will actually take pictures of what they see. They'll put their phones up against the eyepiece. It's tricky, it's not easy, but they'll take a picture of what they see, and that sometimes helps them. And then they take their phone to their table and they do more drawing. It's nice because we don't have a microscope for everybody, so if you think that that's something that could help you, you are more than welcome to do that, okay? So there I am looking at medium power at this specimen. Most of the time, we're going to ask you to draw in high power. So now we're going to go one more time, turn our revolving nose piece to our highest power. That's our blue objective. It has a power of 40. So once again, 10 times 40. Total magnification is 400. So now when I'm looking through the eyepiece, I'm looking at a magnification of 400 times the naked eye. I'm going to move him just ever so slightly. Another trick, and this is going to be something you see on your lab next class. When you are looking through this, it's almost like a series of mirrors. When you're looking through and you're moving the slide, if you move the slide to the left, the image in here moves to the right. And if you move this slide to the right, the image in here moves to the left. You move it forward, the image here moves backwards. So everything's opposite, so you gotta be careful. Whenever you're trying to make adjustments with your slide on the stage while you're looking, very, very small. Okay, you're looking at a field of view here of about 2 centimeters, so it's not very much. Okay, so I have my specimen, it's dead center because I made sure that it was dead center when I started here. I'm going to do a little bit of adjustment. Again, I'm using the fine adjustment knob, making sure that it's nice and clear. For this organism, there's definitely a lot of layers to it, so I can see that as I do this, some parts of the cell are blurry and some are more fine-tuned and crisp and clear. So I will just continue to play with that as I'm doing my drawing and making different parts of the cell, okay? So again, you wanna really take your time on that. Your drawings are really important. It doesn't matter if you're a good artist, it matters how much time you take. Always use a pencil when you're doing your drawings, and I do need your drawings to be good. I need to be able to look at your drawing and know exactly what I'm seeing, okay? Once you are finished with your microscope slide, because again, I told you there's gonna be 6 that you're looking at and drawing. Again, if you take a picture, that would help so that you can free up the microscope. But once you're finished with the microscope, first thing you do, turn this back to the lowest power, the scanning objective. Oops, I didn't do that. Scanning objective, that's the red one. It's the shortest one, it's easy to see. You turn it there, you're going to take your slide clips off the slide, you're gonna remove the slide, you're not gonna lay it on the counter next to your microscope, you're going to put it away, okay? Put it away, and then you're going to turn off the microscope, okay? If we're done for the day, if you're the last one to use it, you're going to unplug it, you're gonna wrap the cord, and you're gonna put the cover, the plastic bag, on top, okay? Got it? See you next class.